Okay, bio, this is going to be the second part of video 2. So on your table of contents, this is going to look like video 2B, Intro to Punnett Squares / Complete Dominance. In video 2A, I introduce you to some of the vocabulary we're going to use in your Punnett squares. And in video 2B, I'm going to actually walk you through how to complete the Punnett square. Now at the end of video 2A, I just barely stopped on this term right here, hybrid. So a hybrid is another way to say heterozygous. A hybrid is when you have both forms of an allele. You have the dominant and the recessive. Usually, if you have the dominant and the recessive, only the dominant is going to be shown if it's complete dominance, which I'm going to teach you about later. If it helps you to remember hybrid, think of animals that are hybrids, like a liger, a lion and a tiger, or a mule is a horse and a donkey. We'll talk about more hybrids in class if that helps you to remember. But when we say a hybrid trait, we're just talking about one that has both the dominant and the recessive. Now this one, I know I keep going back to height in pea plants. This one has a hybrid or heterozygous genotype, but the phenotype is it's tall. So think about why that is. If it's got this short gene, why does it show as tall? Because the tall allele is dominant. The Punnett squares that I'm going to teach you how to do, most of them are called monohybrid crosses. Think about the prefix mono. What does the prefix mono mean? It means one. We're looking at only one trait and only 2, possibly 3 forms of that one trait. Monohybrid cross is going to be your Punnett square that just has the 4 boxes. Example, height in pea plants. When we're crossing those big T's and little t's, we're only looking at height of pea plants. We're not also adding color or color of pod. That, if we get time later, is called a dihybrid cross, and I'll teach you how to do that if we get some time later. The first Punnett square I'm going to teach you how to do is called complete dominance. One of your forms of the gene is totally dominant over the other one. I say one allele. Recessive, you only see if that bully dominant trait is not there. This is complete dominance. One is totally dominant over the other one. Back to my pea plants and height. Tall is dominant over short. If you inherit 2 dominant alleles, this would be called homozygous dominant. The phenotype is going to be tall. If your genotype is heterozygous, the tall still wins, hiding a short gene, still the phenotype is going to be tall. If you have 2 lowercase or your recessive trait, that's the only time that a plant would show up as short. So you only see the recessive allele if there is no dominant there to mask it. And here is your Punnett square example of complete dominance. So we have parents. Let's consider, this is one parent right up here. Let's say this is the mommy plant, okay? Mommy plant is homozygous dominant, so 2 dominant alleles. Mommy is definitely a tall plant. Daddy plant is heterozygous. Daddy plant still looks tall, but is hiding one short gene. So looking in here, these are our possible offspring. This does not mean that if I make 4 plants, they're going to look just like this. This is just the possibility out of crossing these 2. First one uppercase T came from this parent. The other uppercase T came from this parent. That's why you guys can see I've got the blue here for the parent on the side and the yellow for the parent on the top. You can see that each offspring is made out of half of the mommy plant, half of the daddy plant. Okay? There is no possibility for 2 lowercase t's here. There is 0 probability of that because I don't have 2 lowercase t's to give. Remember, you have to get 1 from this parent and 1 from this parent. Now, one thing you need to be able to do out of a Punnett square is to figure out percentages of genotypes and phenotypes. You have to show me all the possibilities, even if it's a 0%. So genotypes, this is the genes. I can't see your genes. I can see your phenotype. I can see the actual physical result. But if I have the genotypes of the parents, I can figure out the possible genotypes of the kids. So here we had, think of each one of these squares as 25%. If it helps you remember 4 quarters and $1, 25, 25, 25, 25 makes 100%. Okay, 25 cents, 25 cents, 25 cents, 25 cents. So we have 25 plus 25, 50% of our offspring would, if we were to breed like 100 plants, we would expect at least 50 of them to be this genotype, homozygous dominant. Make sure you write these little teeny tiny ones next to it. Another 50% would possibly be heterozygous. Remember, this is the probability. I have 0 probability of 2 lowercase t's out of this. If I were to breed a plant that had 2 lowercase t's, I would have to go back and check the genotypes of my parents. That's not possible. Okay, 2 lowercase g's or 2 lowercase t's, sorry guys, or a short plant, not possible out of this cross. 100% of my offspring are going to be tall because they all have uppercase t's. Uppercase T wins. This is complete dominance. Anytime you have an uppercase, it's always the one that's going to win. It's completely dominant over the other. And here is your summary. This is the summary for 2A and 2B. First one says, what does a Punnett square tell you? And then your second question is, I want you to do a Punnett square just like the one you saw on the slide before this. So cross a true breeding tall plant with a homozygous short plant. So using uppercase T's and lowercase t's, give me those genotype and phenotype percentages. I want to see something like this. All right, I want to see the full Punnett square and all the genotypes and phenotypes. Even if it's 0%, I want to see the possible genotypes and phenotypes out of that trait, not necessarily out of that cross. All right, that is it for 2B. You guys are all done.